Thank you for joining us today. I'm Shannon O'Fallon, Program Manager for Team Amazon Fred Hutch Obliteride. Obliteride is a fundraiser that fuels research at Fred Hutch. Held every year in August, participants ride one of the 25, 50, or 100-mile bike routes or join the 5K walk or run. And this year, due to the pandemic, Obliteride was a virtual event with thousands of people joining worldwide. And this year, attendees could pick their activity that they love. They could ride, walk, run, or even bake cakes or do yoga. They also had fun at the online events and also raised millions of dollars for both cancer and COVID-19 research. I became involved with the Obliteride program back in 2017 when I was going through cancer treatment at the Seattle Cancer Care Alliance. I started slow. I was fundraising at first because of the physical limitations from cancer, to then participating in the 5K walk and acting as the volunteer team captain for Amazon's Obliteride team for the past 2 years. In my new role as program manager for Team Amazon Fred Hutch Obliteride, I will be responsible for the global effort to help accelerate Fred Hutch's mission of curing cancer by 2025. I will take what we've learned over the past few years and put a formal structure in place to build repeatable mechanisms that can scale not only for the Hutch, but for similar customers with impact-focused missions that benefit society in some type of way. I've always said that I can't change the fact that I had cancer, but hopefully I can bring about change because I had it. I choose to support the Hutch because of their research. I truly believe that I'm here today because of the amazing treatment and care that I received. The Hutch is a pioneer in cancer research, and they continue to take risks and innovate on life-saving research. Amazon partners with Fred Hutch in other ways to end cancer, and this year, COVID-19. Currently, Amazon is supporting a Fred Hutch study called COVID Watch, which is following people with the virus to determine how long they can spread it to others and if it's possible to be reinfected. Many of you joined the June 15th Fishbowl with experts from our partner and Seattle neighbor, the Fred Hutchinson Cancer Research Center. It was so well received that we brought back Dr. Tom Lynch and 4 more researchers who work on COVID-19 to give us an update. It is my absolute pleasure to begin the program by introducing our guests. Dr. Tom Lynch is the president and director of Fred Hutch and holds the Raisbeck Endowed Chair. He is a world-renowned scientist who played a pioneering role in developing precision medicine. Prior to joining the Hutch this past February, Tom held leadership positions at the Yale Cancer Center, Mass General, and Bristol Myers Squibb, and was also a professor at the medical schools of both Harvard and Yale. Dr. Jim Bunyaratana-Kornkit is a physician and scientist who studies the immune system and respiratory viral infection. Since the pandemic began, he has been studying why people respond differently to the coronavirus and how scientists can use that information to create better treatments and vaccines. He's also playing a very important role in COVID Watch. Dr. Holly James is a biostatistician who helps to design and analyze vaccine studies for the HIV Vaccine Trials Network. She is using her expertise to coordinate 5 COVID-19 vaccine trials involving more than 150,000 volunteers at more than 100 trial sites in the US and worldwide. Dr. Catherine Liu is a physician who specializes in the prevention and treatment of infectious diseases that affect patients with weakened immune systems. For the last 7 months, she has been working to make sure that these patients who are particularly vulnerable to COVID-19 are able to safely receive care at medical facilities like Seattle Cancer Care Alliance. And finally, Dr. Josh Schiffer is an infectious disease physician and mathematical modeler who specializes in viruses like HIV, herpes, and now COVID-19. His particular area of expertise is developing models to optimize treatments. When not treating patients or devising new treatment regimens, he's been educating the public about COVID-19 in YouTube conversations with actor John McHale. Thank you all for being here, and for some of you wearing your Obliteride jerseys. Tom, I understand this was your very first Obliteride. What was the experience like for you?
Shannon, thanks so much. And I want to say I did not do Obliteride wearing a tie and a suit. So I want to tell you, I did have my Obliteride shirt ready. And I want to thank Amazon, all the employees of Amazon who participated in Obliteride, all the participants from the Hutch. We had more than 3,000 people from every state and continent. We even had people in Antarctica who were riding Obliteride for us. We raised more than $3 million. And it really was an extraordinary way to bring together community. And Shannon, as a cancer survivor, I think you probably more than anyone understand how critical community is and friendship and connections are to getting people through a cancer experience.
Yes, I definitely do. And I'm really excited that we're here today and going to have another discussion. It seems like June was, months ago. Can you share some of the significant developments at Fred Hutch since you were here last?
So obviously, lots of things happen. So I want to say one thing. The Fred Hutch, we work really, really hard on finding cures for cancer. It's a big part of what we do at the Fred Hutch. I think that's really important to keep reminding people that. However, we do have an incredible group of people who are working on viruses. For years, HIV was a big, big focus and remains a big, big focus of the Fred Hutch. But as we talked about in June, one of the things I'm very proud of was how many of our scientists are able to be working on the coronavirus and COVID-19. It's really absolutely remarkable. And two big things I think have happened since June that I think we're going to enjoy talking about today. First, we are launching a number of vaccine trials through the Coronavirus Prevention Network. This is all part of Operation Warp Speed. We are working extremely hard to be able to bring vaccine trials to maturity so we can find out if the vaccines are safe and effective. And you just have to pick up any newspaper or watch any television news program to understand how the public is desperately interested in the safety and efficacy of vaccines for coronavirus. And it's something we're working on very hard. Second thing, we opened a COVID-19 clinical research center. I shouldn't say it's opened yet. It won't be open for about a week. But this coronavirus COVID-19 research center is going to be an outpatient center. So, one of the issues right now is that many of the drugs and treatments that have been developed, things like dexamethasone and remdesivir, two really effective treatments for COVID-19, have been developed in patients who are hospitalized. And many of those patients have been on ventilators or had severe oxygen requirements or many other problems which complicated their care. And it showed that those treatments actually could make a difference in the inpatient setting. Well, one of the things we learned from cancer is that taking an effective drug and using it early in the treatment of disease can make a big difference. And we know that from cancer. We also know that from viruses. The drug Tamiflu works better when you use it earlier in the course of a disease. So what we need is a place where we can treat people in an experimental clinical trial setting early in the course of their disease. And that's why we launched the COVID-19 Clinical Research Center. We're incredibly excited about that. We're going to be looking at monoclonal antibodies against COVID. We're going to be looking at drugs against COVID. It'll be a very exciting place to be.
Thank you, Tom.
So let's start with some questions for the group. Catherine, you're an extraordinarily accomplished clinician. One of the jobs you do at the Seattle Cancer Care Alliance is you work on infection control and the interplay between infectious disease and patients with cancer. And we heard from Shannon as a cancer survivor how critical it is to support cancer patients during this time. Can you talk a little bit about COVID and cancer and how we're doing trying to meet the needs of our cancer patient population in the face of the COVID pandemic?
Thanks very much, Tom, and to Shannon for sharing your story, to Amazon for having us here today. With Seattle being the initial epicenter of the pandemic here in the United States, we were one of the first cancer centers that was challenged with responding to COVID-19. And I think we've really learned a great deal over these past 7 months or so about how to keep our patients safe while ensuring that they can continue to receive the treatment that they need. And one of the things that we implemented very early on was symptom screening of our patients and employees upon entry into our cancer center. What this allowed for was rapid identification of potentially infected individuals who could be referred for testing and appropriately isolated in the case of a patient or sent home in the case of an employee. We also reinforced a strict stay-at-home when sick policy among our healthcare workers. And we felt that these were critical first steps to limit transmission events and exposures in our clinic. particularly among our highly vulnerable patients. We also continue to emphasize the importance of basic infection prevention strategies, including hand hygiene to our patients, our caregivers, and our staff. And then as data emerged regarding the value of masks in limiting transmission, we implemented universal masking of our patients, caregivers, and employees in the clinic. To the extent possible, we have also attempted to support physical distancing in waiting areas and other areas of the clinic, and have rapidly adopted telehealth and remote work for staff where feasible as strategies to be able to continue to provide care for our patients while limiting exposures. And with all of these strategies in place, we have really been able to bring most of our patients back into the clinic, and we are operating right now at pre-pandemic levels, and while at the same time continuing to ensure that our patients can remain safe while receiving their treatment.
Thank you, Catherine. So Jim, Shannon mentioned COVID Watch. Can you tell us a little about the trial and what are we hoping to learn and how is the trial going?
Thank you, Tom, for asking about COVID Watch. I think this is a vital study being conducted at the Fred Hutch in partnership with Amazon. It has the potential to yield deep insights into how our immune systems respond and protect us from COVID-19. So I'm the physician on the ground for this study. And as a scientist, I also work in the laboratory to analyze some of the specimens that we have collected. So we're leveraging innovative technology to address some of the unanswered questions about COVID-19. For instance, our participants, they swab themselves from the safety and comfort of their own homes and mail back their samples to us to test for the virus that causes COVID-19. Participants are also given a portable device that allows them to self-collect at home a small sample of blood and mail that back to us as well to test for antibodies against the virus. And participants who test positive are utilizing our drive-through tents at the Hutch, where they can see a clinician and provide a larger blood draw for us to analyze the actual cells that respond to the virus and might protect against a second infection. The real power in this study comes from following people over time, from before they acquired the virus to months after their infection. And this information is vital to understanding how pre-existing immunity might affect the course of infection and how the immune response to the virus changes over time. All of this plays into tackling those unanswered questions of how long immunity might last after infection, why some people get sicker than others, and if the other common cold coronaviruses affect the disease course of COVID-19. So we enrolled our first patient back in April, and the study is humming along. We are expanding into central and eastern Washington so that underserved, underrepresented groups outside of the Seattle area also have the opportunity to participate. We really want our findings to be generally applicable to all people. And if I may make one side note, as the study physician for COVID Watch, I'm the person who calls participants when they test positive. And anecdotally, I've found that there are more and more people who have become a little bit lax with mask wearing. I totally get this. I mean, we've been in this pandemic for so long now, and it can get tiring and we are getting fatigued, but I do want to emphasize the importance of wearing masks, both for protecting yourself and protecting others. And hopefully with our experience, I can encourage those who are listening to stay vigilant with your mask wearing.
Jim, thank you for that note. That's incredibly important. One quick question I have for you. So you allow people to self-collect. I was talking to my son who's in a study where they're doing a comparison between self-collected versus nurse-collected specimens. Do you think there's a difference?
Thank you so much, Tom. That is part of our study, actually, to compare self-collection with clinician-collected samples. That I think the jury is still out. There's some data that shows that self-collection works pretty well, but not quite as good as a clinician doing a deep nasal pharyngeal swab where they really get back there. Okay. It's good. It's still pretty good, but it's maybe not as good. So thanks, Tom, for that question. Great question.
To say the testing I've had has always had with a clinician doing a deep nasal pharyngeal swab and getting back there. So I would love to see that maybe some of these less invasive tests could be helpful. So Holly, you're working on the coronavirus prevention network. Again, something of enormous import. I mean, you think how critical your work is to the future of how we're doing with this pandemic. It really is extraordinary. Can you tell us a little bit about what you're doing and what's the status of the vaccine studies at this point?
Thanks, Tom, and thanks to everyone for organizing this event. I'm glad to be here. Yes, the Coronavirus Prevention Network is a large multinational effort engaging multiple previously constituted networks of investigators that work together for studies of other pathogens that have been kind of reconstituted and brought together for the study of COVID vaccine research. And there are currently 5 efficacy trials of candidate COVID-19 vaccines that are slated to get underway in terms of evaluating their efficacy under the umbrella of this network, and partially funded by the U.S. government. There is, as folks have heard and seen in the media, there's one efficacy trial that's underway that started in late July. There's another that started very soon thereafter, and that's currently in a pause. And then there are 3 more that are slated to start sometime in the next few months. My role is to I'm a biostatistician by training, and I think about how do we design these trials, at a very basic level. What are the populations of individuals who should be recruited to these trials? Where should the clinical trial sites be located? And that requires some thinking and anticipation about, where the epidemic is going to go, where it's going to take us over the next few months. How many participants do we need to enroll, and how are we going to measure, importantly, the safety of these vaccines and establish the safety, robust safety data on their safety, and how are we going to measure the efficacy of these vaccines, and how long is that going to take, and what are we going to do if it takes longer than expected to get an answer?
Yeah, and I think one of the things we saw this week was, I can't remember if it was the Moderna vaccine or the Pfizer vaccine, where they've completed the initial accrual of 29,000, and then decided to go to 44,000 to get a more diverse patient population. Tell us why you would do that. What's the advantage of increasing your accrual to diversify the patient population?
Yeah, thanks. So primarily, a key reason that we're really concerned about the diversity of these populations in these vaccine trials is because of the disproportionate impact of COVID on, in particular, minority populations. So we've seen that the impact is disproportionately affecting those populations, and so those populations have great potential to benefit from an effective vaccine. And so we really want to make sure that we're including in substantial numbers individuals from those populations, so that we can again definitively establish the safety and efficacy of those vaccines in those populations that have the most potential to benefit from them.
Great, thank you. So next we'll move to Josh Schiffer. Josh, you specialize in building models that look at the progression of viruses in infected people. What do the models tell you so far about the effectiveness of COVID-19? We hear a lot about the super spreader events, and we hear a lot about how in certain circumstances, one person can spread to many, many people, and others, one person might not spread to anybody. What does the modeling tell you so far? And what have you learned over the past 3 to 6 months about COVID-19?
Thanks, Tom, and thanks, Amazon. It's great to be here. So our group was very interested in this observation that's been made in many countries across the world, which is that SARS-CoV-2, actually the majority of people who get it don't infect anybody, in the neighborhood of 70 to 80%, but that 10% of people are responsible for about 80% of infections. And the way that plays out is that you actually have a decent number of people who infect anywhere from 5 to 10 people, and then you have a tiny minority of people, and you often read about these situations on fishing boats and in meatpacking plants and in fraternities, in prisons, where one person has the potential to infect dozens of people and potentially even over 100 people. And our first thought was that influenza, which in some ways is comparable, does not behave this way. It rarely infects more than there's rarely a situation where one person infects more than 10 people. And so the way we sought to address this was to think of the reasons why. And the first obvious reason that came to mind was that people with SARS-CoV-2 actually shed for much longer. And so one possible explanation is that because they're shedding for many weeks, in many cases versus 1 week, maybe they're infecting people over that whole period of time. That turned out not to be the case. What turned out to be the case is probably best described with, if I make up a situation where you have the sort of stereotypical crowded, poorly ventilated space with lots of people, and you were to have a person with influenza at their maximal transmissibility, so shedding at their highest viral load, and also a person with COVID also shedding at their highest viral load, walk into that environment, the person with COVID would infect many, many more people than the person with influenza. And so our model really contributed to a complementary literature that suggests that the virus, at least relative to influenza and other respiratory viruses, can aerosolize and can spread over a wider area, particularly in poorly ventilated areas. And the adjunctive piece to that was that it's really people who are potential super spreaders by virtue of being essential workers who must go to a crowded environment every day are really important targets for prevention, be it by vaccines, be it by masking. Because if you can stop these events, you bring the so-called reproductive number well under 1, and the ability of the virus to grow in the community and spread is eliminated. So it's not only an interesting observation, but it's potentially a very important observation for how you target prevention in the community.
That's fascinating. What have we learned, Josh, about transmission in some of the more public events? For example, some of the protests here in the U.S., sporting events around the world, political rallies around the world. Have these areas turned out to be big spreading events? Do we not know? What do we know about some of these larger events?
Yeah, I think the first piece to acknowledge is that it is very hard to deconstruct how important these events are. I think in a general sense, we've learned very importantly that being outside decreases the risk of transmission dramatically. And I think it would take a very special set of circumstances to have a super spreading event outside. It's not to say that transmission can't occur outside, but it's less likely. But it is very difficult to deconstruct. For example, there was a recent article that was then quickly debunked about the Sturgis motorcycle rally contributing to tens of thousands of cases across the country. And that actually is not out of the question, but it's just incredibly difficult to prove because of many factors, one of which having to do with some of the work that Trevor Bedford is doing with the viral evolution and the virus simply not mutating quickly enough to deconstruct chains of transmission over that short of a time frame. But I think we know, although it's hard to get precisely at your answer, we certainly know what it takes to really stem transmission, and that is to avoid crowded events indoors whenever possible, and certainly to wear effective masks and to comply with those masks whenever possible. I think we have those tools in hand, and we've had those tools in hand for a long time, and that really can get us to a place where we can, if really the majority of the population were compliant, where we could get to a place where we were relatively safe prior to implementation of a vaccine.
Okay. So, Shannon, you can probably imagine, I don't know if this is true at Amazon, but when we have our town halls at the Hutch and we talk about COVID, by far the most number of questions we get is about masks. And I wonder if that's the case at Amazon as well, since people want to know what type of mask to wear, how to wear masks, how effective are masks. And so, Catherine, as someone who thinks about infection prevention, tell us a little bit about the importance of masks. And what about these exhalation valves or vents that are present on masks? Does that change the effectiveness? Is it something that is the mask still effective in that setting? And where do you stand on recommendations for mask wearing?
Thanks very much, Tom, for this question. So the main purpose of masks in this pandemic is really to serve as a barrier to prevent respiratory droplets from being transmitted from one person to another. And so with respect to these masks with exhalation valves or vents, these are really not effective because they have essentially a hole that allows for air to be exhaled and thus no longer serve as a barrier. And these masks, I think, were really designed for use in construction work, as the valve is thought to make the mask more breathable by allowing the air to escape from the mask. But the same feature of the valve essentially defeats the goal of source control and preventing viral transmission, which is really what we're aiming for. With this pandemic. And so we have recommended against use of masks with exhalation valves or vents in our clinic. And this is consistent with the guidelines by the CDC as well. I think that when thinking about masks, there are really a lot of choices now as we've moved initially from homemade masks to innumerable options now, as I think that the cloth-based mask industry has rapidly exploded over the last couple of months. And this has, I think as Josh mentioned, really become one of the cornerstones of prevention of transmission, both in our communities as well as our hospitals. And I think there's just a couple of things to keep in mind when you're choosing a mask. Ideally, the mask should have at least 2 or more layers, and importantly, should fit. I think this is probably one of the most important things. It really should fit appropriately. It should completely cover your nose or mouth. Sometimes we see people wearing the mask, hanging over their neck or below the nose. That's really not the proper way to wear a mask. And we want to make sure that the mask fits snugly without any gaps around your face, just to ensure, as tight of a seal as possible. So I think from our perspective, and certainly in the clinic, this has really been a key part of us allowing us to be able to continue clinical care for our patients. And I think in the community as well, I think you can think about masks as one of, again, the key strategies in trying to limit transmission. It's part of the 3 Ws. If those of you are not familiar with the 3 Ws of trying to prevent transmission include wearing a mask, washing your hands, and wash your distance. And so I think, as others have mentioned, we continue to strongly recommend mask usage as one of the key strategies to limit transmission in our communities. And I think maybe I'll hand this off to Josh now to maybe talk about some of his latest research on masks and their impact on limiting transmission events.
Yeah, great. Thank you, Catherine. And I really second everything you said about the proper use of masks and selection of types of masks for prevention. Unfortunately, driving around the city, I see, as Jim mentioned, inappropriate use of masks quite frequently. And we always have, they're not great, but we always have to remind ourselves how important that is. So my background prior to COVID was as an STD researcher. And so the mask is like a barrier method. And so I immediately thought of condoms as the other barrier method to prevent infections. And masks are funny because compared head to head at the individual level, they're not as good as condoms. They're not guaranteed to protect an individual. But if both the potential transmitter and the exposed person are wearing a mask, the degree of protection is amplified. And if this is averaged across the population, the number of infections and cases and deaths that can be prevented is really profound. And in fact, even a slight uptick in the use of masks at the population level in the US, I personally think would get us at a reproductive number where it would be safe to do things like open schools. And so it's really a dial that we can turn right now to get us in a safer place where we're not flirting with new outbreaks and new periods of intense pressure on emergency departments and ICUs, and unfortunately deaths. So it's really the key tool we have, and just improving a little bit across the board would have really massive impact at the population level.
Okay. Thanks, Josh. So I'm now going to ask Holly the hardest question. I asked Catherine the most common question, which is about masks. But Holly, you're definitely going to get the hardest question, which we've seen this week. We've seen former Vice President Biden. We've seen President Trump. We have seen Dr. Redfield. We've seen Dr. Fauci. We've seen a million opinions about when the vaccines are going to be ready. So from your perspective, If you could give us your insight in when you think the vaccines might be ready. And then I think the second question people really want to know about is, how are you evaluating these for safety and efficacy? What defines a safe vaccine? What defines an effective vaccine?
Thanks. So in terms of timeline, I would say the biggest answer is we don't know. It depends. So the trials have all been designed to optimize the likelihood of getting as fast an answer as possible while ensuring that we can collect adequate safety and efficacy data. And so what that has meant is that they've been designed to under the best information we have at the time at the trial design to provide an answer in about 6 months from the time the trial opened. Now, if you look at the time that the first trial opened, that would have put us at about January as the earliest time point at which we would have an efficacy result for the first trial. However, the trials are all what we call, as statisticians, event-driven. So the primary analysis of efficacy will occur when there are enough COVID-19 cases in the trial to provide adequate statistical power to evaluate vaccine efficacy. And because the pandemic is evolving and transmission is impossible to predict, and incidence is impossible to predict precisely in advance. We don't know exactly when that primary analysis will occur, and so it could be sooner, and it could be later, depending on what is going on with transmissions, both in the population as well as in the cohort of participants who are enrolled in these trials. In addition, there's other uncertainty associated with the fact that efficacy and safety are being monitored in real time as these trials are underway. And so it's possible that a trial could reach an efficacy signal earlier than anticipated, but pretty unlikely. So it's possible that an efficacy result could be delivered 1 to 2 months earlier than when it was anticipated. So the short answer is that it depends, and we don't know. It depends on how the pandemic evolves, and it depends on how well these vaccines work. But all efforts are being made to deliver, an answer in as timely a fashion as possible. And in terms of how we're evaluating safety and efficacy, I would say that the short answer on safety is that we're evaluating it in the same fashion and in the same rigor that all other vaccines have been developed and licensed in the U.S. and internationally. In particular, we look at, reactogenicity in terms of these vaccines. We look at adverse events that occur amongst the trial participants, and those are monitored both, in the near term around the time of the vaccinations, as well as over long-term follow-up. So the participants in these trials will be followed for 2 years to really comprehensively characterize what happens to these people in terms of incidence of COVID, as well as other potentially associated safety events. In terms of efficacy, so the short answer on efficacy is that we will at the end of the trial, at the time of the primary analysis, will compare the rate of COVID-19 in the participants who will have participants enrolled. Some of them will be randomized to receive a candidate vaccine, and some of them will be randomized to receive a placebo that looks and seems in all fashion similar to vaccines, so that the participants are blinded and they don't know which they're receiving. And at the end of the primary analysis, we'll compare the rate of COVID-19 in the participants who are randomized to receive the vaccine compared to that in the participants who are randomized to receive the placebo. And we hope to see that there's a considerable reduction in the incidence of COVID-19 in those who receive the vaccine relative to those who receive the placebo.
So Holly, one question that could be asked, and has actually been asked to me this week, is we've heard about one side effect in one patient in England who was on the AstraZeneca vaccine. Does that mean that vaccine is not safe? What does that mean?
It means that there's really rigorous safety monitoring going on in these trials. So that event I can't comment on in particular. I don't have the information, but I could say that it's being monitored by each of these trials has a data safety and monitoring board that's built in that is an outside team of experts, including clinicians, biostatisticians, ethicists, community advisory community members who review in real time the data that accrue on each of these vaccines, on the trials individually as well as in aggregate among other trials that are related and that are relevant, that are in the field. And those committees really are responsible for making the determination as to whether or not that particular safety event and any others that might occur in these trials could potentially be related to the vaccine, or could be a pre-existing or unrelated condition that accrues in these trials.
And I would say one thing, Shannon, for the employees of Amazon, having done this kind of work for cancer drugs, I can tell you it's taken extremely seriously. And we really do look at the side effects in the treatment group versus the side effects in the placebo group and try to determine, really what's going on there. So Josh, one of the questions is with it, the virus mutates, we know it mutates. Some people say it mutates relatively slowly. Others people say it's been mutating faster. What implications does this have about efficacy of vaccines? Is the virus mutating so fast that maybe a vaccine won't be efficacious?
Yeah, so I would first of all second exactly what Holly said, which is that we don't know yet whether the vaccine will be efficacious or whether some vaccines will be more efficacious than others. My tentative opinion on this comes from following Trevor Bedford's work very closely at the Hutch, which is that his opinion is that the virus does not appear to be mutating quickly enough that it would be a sufficient explanation for vaccine failure. On the other hand, it does appear to be mutating quickly enough that he would not be surprised if we do develop an effective vaccine that we need to supplement it periodically. Whether that is every year or every 3 years is less clear, but whether we need to get an updated vaccine, much like we would do for influenza, to account for the mutation of the rate of the virus. But he doesn't seem to think that it should impact the trials that are being done right now. But that is a tentative answer, and we will obviously have to wait and see the data.
Yeah, and Holly, along similar question to what I just asked Josh is about, do you think the vaccine that we get initially will be the vaccine we use forever? Is it a chance that there'll be different types of vaccines that will come out into use?
It's speculation at this point. I think it's like Josh said. It's quite like these are the first generation of these vaccines, we will make every effort to study all aspects of efficacy, and in particular, another aspect is how durable will the immune responses be that are generated by these vaccines, and so we'll make sure to capture that. We'll have an initial assessment of efficacy, and then we'll continue to follow these participants, as I say, for 2 years to see whether the immune responses that these vaccines generate wane, and whether or not there's continues to be efficacy over a longer time period, or whether or not, when moved into the field, these vaccines need to be modified to induce more durable immune responses, or booster vaccinations are needed. Speculation, but probably these are first generation, and
that's interesting. I had a discussion with Larry Corey, who all of you work with extremely closely, and Larry was telling me, he said, Tom, think about it. We've got to vaccinate the entire world to make this effective. And he said it's very, very, at least his opinion is, he said it was very likely we're going to need more than one vaccine, that there'll be more than one vaccine that emerges. I also think we have to think about different patient populations. You've got elderly patients, you've got immune compromised patients, you've got children, you've got people with other conditions. How, will every vaccine work as well in every single population? That's another big question. As a clinician, I'd like to see coming out of the trials to try to get a sense of that. So I'm going to ask a question to Jim, and these questions we're answering now come in from our colleagues at Amazon who are thinking about this problem. So Jim, tell us a little bit about that. You're a clinician. We see some patients with coronavirus, COVID-19, who barely have any illness at all, and then you've got others who are intubated with near death, and others who die. Why such a wide variety of disease that you see from one virus?
Thanks, Tom. That's a great question, and thanks to whoever at Amazon asked that. So yeah, as you alluded to, the spectrum of disease is so wide from person to person with COVID-19. Some are asymptomatic, some have mild cold symptoms, some have a more severe lung infection, some are developing respiratory failure and dying. We've had rare reports of children developing an inflammatory reaction affecting multiple organs. And we may talk about this later. We're getting reports of these long haulers as well, this phenomenon of patients having chronic symptoms. And right now, we do know that several clinical factors play into which patients are at greater risk for developing severe disease. And these kind of make sense, like older age, having other medical diseases involving the lungs, having cancer, as Catherine talked about earlier, kidney disease or diabetes, for instance. The immune response to the virus likely is contributing to why some patients get severely sick. This is true for other viral infections as well, where the immune system overreacts to the virus to try to keep it under control, but causes significant collateral damage to the body. And you've probably heard of the studies that found that a commonly available steroid that dampens the immune response could save lives in people who had severe COVID-19.
And Jim, can you tell us a little bit about the plasma treatments that the FDA has given an emergency authorization for? Why do they work? And if the plasma treatments get you so far, do you think the monoclonal antibodies that are sort of based on the same kind of principle might get us even further? Tell us a little bit about that line of research.
Yeah, great question, Tom. I'm really excited about this avenue because that's area that I'm also doing research in. So starting back in April, I was involved in collecting plasma here in Seattle. And in Seattle, we had very generous donors. And so what is convalescent plasma? Plasma is a blood product that contains antibodies. So after an infection, some people will produce antibodies that can protect against COVID-19. And if you collect plasma from those donors who have recovered, you could transfuse that to other patients with active COVID-19 as a form of treatment. And this has been done in the past for other infections like diphtheria, measles, flu, Ebola, and even the first SARS virus. It's an attractive option because plasma during a pandemic is readily available. And you probably are aware of that recent study that was conducted here in the United States that reported fewer deaths in patients hospitalized with COVID-19 if they received convalescent plasma earlier and if the product contained higher levels of antibodies against the virus. And that rationale makes sense. With viral infections, like you said, Tom, just like cancer, you want to treat early to get the biggest bang for your buck, the greatest chance for success. If you're too late, cat's out of the bag, the horse is out of the barn. But keep in mind that study was not a randomized controlled trial, which is generally the gold standard for getting FDA approval. But despite that, the findings were really promising. And so the FDA did grant emergency use authorization for that product. Now, the downside of convalescent plasma is you're getting each product from a different donor, and there's immense variability that comes with that. And screening each donor is laborious and not trivial. So I think the future for antibody therapy is in these monoclonal antibodies, which are more homogeneous, standardized preparations of antibodies. And the Hutch has extensive experience with monoclonal antibodies, both as immunotherapy for cancer and as potential treatments against HIV. So we're hoping to study antibody-based therapies both as prevention and treatment at our facilities here at the Hutch. And maybe this is an opportunity I can pass on to Josh to give him a chance to talk about the research being planned at the COVID Clinical Research Center.
Thanks, Jim. It's now Tom's mentioned and Jim have both mentioned that treating early has theoretical advantages, and I just want to touch on that a bit more. The data from other viral infections, including influenza, HIV, and Ebola, is very clear, and it's also intuitive. I think the way you can conceptualize severe COVID would be as an equivalent of the forest fire situation in the West right now. Multiple fires, large fires in multiple organs. That's intuitively much harder to treat than a single fire that is contained in a small environment, which is how this infection starts in everybody. And so the goal of the center is to involve ourselves in studies that address that very early infection when the fire is small and the damage is limited. there are all sorts of strategies we can use, and we're looking at monoclonals in certain studies, but we're also looking at remdesivir, and also really keyed in on trying to find oral agents that people could take at home and even have mailed to their home. And one of the really exciting things that we're hoping to initiate in the next month are trials where we actually mail everything to the participants' homes, so that all of our interactions with the participants are online, making life easier for the participants and also limiting the interaction between people who are potentially contagious and staff. And we're really hoping to mimic the studies that have been done out of the University of Washington and University of Minnesota with hydroxychloroquine, and to do this on a national level to really chase the virus where it is. And so this center has really given us the opportunity to think about how best to prevent hospitalization and death, and also to really think about innovative study designs that I think may ultimately be relevant for other infectious diseases and cancers as well.
So thank you, Josh. So Catherine, I'm going to give you a question that matters to me. It also came in from the Amazon employees, which is this issue of college students. So I have a son who's a junior in college. He goes to Colby College in Maine, and they are testing every other day at his college. And they've done 25,000 tests already. And they're doing an intensive testing. They're doing it as part of a study through the Broad Institute in Boston. Not every college is testing that intensely. And they have restrictions as well. They can't have more than 10 people ever gather. And my son said that to the best of his ability, he thinks that many of the students there are following that. Yet we hear stories of hundreds of kids still gathering in some settings. What do you think about colleges? Is that a unique concern? Because again, Josh talked about indoor spread and people coming together. Just think back when you were in college. Maybe your college was not crazy, but there's a lot of things that happen in colleges that are a little bit harder to control. What do you think?
Thanks, Tom. Really excellent and tough questions, I think, that have really emerged over the last couple of weeks as colleges have reopened. And I think clearly, as I think we've all seen in the media, that colleges have emerged as one of the hotspots of the pandemic. A lot of clusters and outbreaks have been reported in almost every single state. And I think one of the, I think what we've seen with respect to the different universities around the country is that there has been quite a variability in approaches to the way that they have reopened. Some of them have started initially 100% with in-person learning, and then learned quickly that they had to shift to remote learning as outbreaks occurred. Others have taken a hybrid approach, and then while others have started out with remote learning from the get-go. And, I think that there's a couple of, I think we're still learning about what the optimal strategies are to really bring our students back to school this fall. And I think there's a couple of things that probably we should be thinking about. And I think, as you mentioned, the rigorous testing approach that you're describing at your son's college, I think that's a critical piece. Obviously, not every college has the capacity to be able to do that kind of testing, but I think that being able to test repeatedly and frequently in a setting where you have a congregate environment, even in settings where colleges are opening with nearly 100% remote learning, many of these students are still on campus. They're living in campus housing, so they are still in congregate environments. And so having a rigorous testing strategy, I think, is really going to be key to trying to limit transmission events, in addition to contact tracing. And quarantining students if they're positive. And obviously these pose tremendous challenges for universities that may not have those type of resources to do so. I think the other, I think just looking at some of the experiences here at University of Washington, some of the strategies I think they've been taking have been similar. They are also offering a robust testing a program that's being done in collaboration with the Seattle Flu Study, the SCAN program. They are, again, in addition to testing symptomatic individuals, they're offering and strongly recommending testing of all students and faculty prior to return to campus. They also plan for weekly testing of a subset of the community. Mask wearing, as we've talked about, is going to be critically important and should be required. again, in all indoor spaces or in environments where physical distancing is not possible. I think there's been some variability across the country with respect to that requirement at different universities. And I think that, again, while, I think a lot of schools have had to move to remote learning again because of the outbreaks that have occurred. I think one of the challenges, even with moving to remote learning, is that the universities oftentimes don't have control over activities that occur outside of the classroom. So, Friday night parties, college students congregating together, doing what college students do. And I think that's really been a challenge in terms of universities not necessarily having oversight, for example, in Greek housing, off-campus housing. Some of them, I think, have actually resorted to some more draconian measures, including suspending students. Though I think in some ways these measures can be counterproductive, as sending these students home may actually mean sending students back to their home communities and potentially infecting those around them. So these are all challenges I think that the colleges are facing. There are, I think, a couple of other mitigation strategies, including to beyond what I've mentioned before, in addition to testing and mask requirements. I think that there are things that colleges can consider that they do have control over. During the move-in period, they might consider creating sort of staggered windows of time for the move-in, limiting the number of individuals who can be there during that process. And I think, as you mentioned, Tom, there have been efforts to try to limit the number of people that can be in any common areas, limiting capacity in bathrooms and in rooms. Again, these can be difficult, though, I think, to reinforce. Having it's difficult, I think, to have to make sure that everybody's doing what they should be doing, and that requires resources as well. I know one of the challenges that I think, again, as I mentioned, testing is critically important, and I think probably ideally should be mandatory. I think at UW, it was difficult to make this from a legal perspective mandatory, so it is currently a voluntary process here, but it's being strongly encouraged and recommended for all students who are coming back onto campus.
And I think, Catherine, we've got some good examples where aggressive testing and mask use seem to be helping. I mean, I've been very impressed by the NBA and the WNBA, how both those leagues have been able to do pretty darn well in terms of reopening. And my hope is we'll see that with the remainder of the sports leagues, that they'll be able to do that pretty effectively. The WNBA being the first example, then the NBA following very closely after that, and having, again, done quite well. So, Josh, I'm going to give you the last question, and this is a sort of a philosophical one for you to think about. Will COVID-19 ever go away?
I don't think the virus will go away completely. I think it's likely to remain what we call an endemic virus, where it's present somewhere on the globe for many, many years, and perhaps for the rest of our lives. I do think that its form will change. I think that in all likelihood, we will see many fewer cases as vaccines roll out, and also as the natural number of people who are immune increases. I think that we will probably see limited outbreaks in certain settings, perhaps where vaccination rates are lower. And there is a tendency among many viruses, though not all, to become less pathogenic over time. And so it would not surprise me if this becomes a severe nuisance, but perhaps a severe nuisance with a less high of a case fatality rate. And there already may be a signal that that's happening, though that could be due to many other factors, one of which is masking. I think that what is really important to consider is that this is not our last pandemic. The world has been, the signals have been very intense over the last 20 years that the way that the way of modern life, where people travel very frequently, where people's housing interfaces with the wilderness, it really does predispose to further pandemics. And so what I really hope is that we're more ready in every front next time, in terms of testing, in terms of tracking, in terms of developing treatments much more quickly than we have this time, and being more strategic about our clinical trials. And so I don't think we're going to be out of the woods in our lifetimes as far as other pandemics, and so it's critical that we learn from this one so that the next is the next event is less of an imposition on all of our lives and has such less of a devastating effect globally.
Josh, thank you. And I want to thank everyone for a terrific discussion. I also want to thank again Amazon for their support of the Fred Hutch. I can tell you when I did the Obliteride and I was riding along Lake Washington, one of the best moments was as I was going down the road, there were a group of about 15 Amazon riders who were coming up the other side in perfect, they were sort of in a Peloton formation. So I think it was a wonderful example of how Amazon teamwork can work together to achieve great things, but also how they're doing this for the Hutch made me extremely proud and very happy to be working with you. So Shannon, thank you so much for welcoming us and including us in this Fishbowl program.
Thanks, everyone. And thank you so much to Dr. Tom Lynch and all of the other researchers for their great work that they're doing at the Hutch. If you want to get more involved, you can support the Hutch's research. You can subscribe to get Hutch news alerts or sign up to participate in a vaccine trial at the Hutch. I'd also like to encourage you to get involved in Team Amazon for Obliteride 2021. Amazon has been involved for all 7 years, and in 2020, we turned out our largest Amazon team with 257 members and raised nearly $100,000. We want next year to be even bigger and better, so please go to obliteride.org or reach out to me directly. Thank you so much for joining us today.